science today has become very interdisciplinary as well as multidisciplinary. So for drug discovery, it's truly a combination of many different fields, right? So there's chemistry, there's physics, there's biology, there's medicine, and there's even economics involved in that. So it's really multidisciplinary.
From the heart of The Ohio State University on the Oval, this is Voices of Excellence from the College of Arts and Sciences, with your host, David Staley. Voices focuses on the innovative work of arts and sciences faculty and staff, with departments as wide-ranging as art, astronomy, chemistry and biochemistry, physics, emergent materials, and mathematics and languages, among many others. The college always has something exciting happening. Join us to find out what's new now.
Joining me today in the ASC Marketing and Communications studio is Dehua Pei, professor in the Department of Chemistry and Biochemistry at The Ohio State University College of the Arts and Sciences. His research spans from the mechanistic study of biological phenomena to the development of methodologies to discover novel therapeutic agents and chemical probes. In 2025, he earned the Ohio State University Distinguished Scholar Award. Congratulations, Dr. Pei, and welcome to Voices.
Thank you, David.
Well, I'm very interested in the research that your group undertakes, and I was really struck by this first one because you frame it in the form of a question. How do biomolecules cross the cell membrane? So what's the answer?
So first of all, let me emphasize this is one of the very important questions that have been in the field for quite some time. People actually have been studying this problem for decades in many different fields. So we can kind of take an analogy of, so the human bodies are made of many cells. And we can kind of look at, compare cell to like orange.
Orange?
So orange has a skin. The skin is basically the cell membrane, the equivalent of the cell membrane. So what people have been wondering about for many years is how do molecules cross the membrane or the skin and get inside the cell, which is the internal components of an orange. So for small molecules, it seems very small, like air molecules, they can permeate through the cell membrane or the skin of the orange rather easily and get in. But for larger molecules, like a huge protein, the proteins are like 1,000 times bigger than small molecules, which we normally consider like a size of a water molecule or carbon dioxide, right? So for those very large molecules, how do they cross the cell membrane, get in, or in some cases get out, has been a mystery for quite some time. Now, why is that important? Well, it's important because we know certain types of proteins can get into human cells. For example, bacteria produce a lot of toxic proteins, we call bacterial toxins. They're very large protein molecules, and they can get into our cells and cause diseases, all kinds of bacterial diseases. And there are also viruses. Not too long ago, we had the pandemic, so we know viruses can get into our cells as well. But actually, amazingly, people did not know how these large molecules or molecular assemblies, meaning a lot of molecules bound together, how do they get into the cell and cause diseases? Or in human cells, some of our proteins will actually get out of the cell and perform functions at the extracellular environment. How do they do that? That's actually unknown. And so we become very, very interested in that. And, that's also driven by a practical need. The practical need is that in drug discovery, so for all human history, or, the history of drug industry, people have been developing what is called a small molecules, basically, relatively small organic molecules as drugs. And so we call them sizes of, 500. You use that numbers as a, kind of as a benchmark. Size of 500 or smaller is what they have been working with. And then, about 40 to 50 years ago, we had a biotechnology industry come along. And then people start to use very large molecules with molecular weight of thousands, even tens of thousands, or hundreds of thousands as drugs. But what they have been able to do is, when they use a small molecule, because they're small, they can go into our cells and, and function and then modulate the activity of the cells and act as a drug. But when it comes to large molecules, like proteins, they can't get it in. So all of the drugs we got out of the biotech industry for the last 40 years, they work outside our cells. And so, it would be great if we can take these large molecules into our cells, and then we can open up an entire new space. And so currently, there's a quote in the industry that says 75% of all disease-relevant drug targets are undruggable, meaning undruggable, meaning the entire industry, the pharma industry and the biotech industry, have no solution for 75% of the disease-relevant targets. And so it would be wonderful if we can access to these drug targets which live inside of our cells. If we can do that, then there will be no undruggable target left.
So what is your lab doing to answer this question that others have not done?
Great question. So we kind of stepped back and we said, okay, well, how does nature do it? And right, if we know that nature has produced viruses that can get in, we know that nature has produced bacterial toxins, many of them they can get in. So we thought, okay, well, let's figure out how nature does it, and then we may be able to design something that does even better, and it goes into the cell and do good, but does not do harm. So that's the rationale behind our research. So then, how do you solve a problem that people have been studying for decades, and were not able to solve it? So we went back to the basically the fundamentals and said, okay, let's forget about all biology, how things people have discovered in the past. Let's come back to the fundamental physical chemical principles. What does it take for something to go from one side of a membrane to the other side? Or if we take another analogy, we're sitting in a room, enclosed room right now. I want to get outside the room, but there's no door. There's no, so how do I get through the wall, get to the other side? And importantly, how do I get to the other side without even damaging the wall? So that is the question, we ask ourselves. So what are some of the ways that you might be able to do it? And it's just, don't even worry about whether that's happening in biology. Just physically, look at the physical chemical principles. Is there a way that things can happen that allow a molecule to go from one side to the other side? So then I remember, well, a little activity that kids do, right? Kids blow soap bubbles. We all know that. So what we observe, of course, is if you blow a big soap bubble, which is kind of like the cell membrane.
Right.
And the big bubble sometimes can split into two bubbles, and a small little bubble come out of the big bubble. And then we've also observed that the small bubble actually will pop. So now you think about it, if you have a cell, you have a membrane, if somehow your molecule can bind to the membrane and then promote the formation of a small little bubble coming out of the larger bubble, and then the molecule will be enclosed in the small bubble, and it will separate, and the small bubble pops, and then you have your molecule release on the other side of the membrane, or get out of the cell in this case. And if you force it to get in, it's exactly the same process, just reverse process. So this is, physics or chemistry, right? So we don't know what happens in the cell. I said, well, that makes sense. So let's just look at in the living system, in a human cell, does this happen? And so once you have a hypothesis, you have some ideas of what you're looking for, you just design experiments and look for it. And sure enough, we found that's exactly how things happen over there. And we first demonstrated this with artificial type of molecules called cell-penetrating peptides, which people have been studying for about 40 years as well, and people did not know how they get into the cell. And we showed that's exactly how they cross the cell membrane. And I said, well, if these artificial molecules can do it in the context of human cell, what about naturally occurring proteins like bacterial toxins? We look at that, sure enough, that's how they do it. And then we thought, okay, well, what about viruses? What about all other systems? And then we went to the literature and looked for evidence, right? And then evidence, it turns out to be everywhere. And so now we believe this is a fundamental mechanism in nature that allows molecules to go from one side of the membrane to the other side. And that's probably a universal, we call it unifying mechanism for many, many different systems.
Why did you decide to tackle this problem? I mean, of all the problems that one could tackle, why this one?
Yeah, so that comes back to the need for drug discovery, right? So today, sure, the pharma industry, the biotech industry are doing a great job. They're developing a lot of new drugs each year. But still today, there are many diseases that, that do not have proper treatments. we have not conquered cancer. Alzheimer's is still a big problem. And there are probably somewhere around 6,000, 7,000 genetic diseases. Very few of them have proper treatment. So there are lots of diseases that are currently undruggable. And it's generally believed that if we can take larger molecules like proteins into the cell, that will solve the undruggable problem. So there certainly has been a lot of incentive for figuring out a way to take large molecules like proteins into the cell. And there's a lot of research going on in this area. So we're certainly not the first one to look into this problem. Like I said earlier, this is a very well-known problem for quite some time.
Are you confident, optimistic that you or someone will solve this problem soon enough?
Well, so mechanistically, we feel like we have solved the problem. There's still, obviously, there are a lot of different systems. We have looked at a few systems, and in the systems that we have looked at, the molecules do cross membrane by the mechanism that we discovered. We call it the vesicle budding and collapse mechanism, or the VBC mechanism. And so that seems to be the case. So there's still a lot of work to do for us and for others to look at other systems. So is it a truly universal mechanism or the unifying mechanism for many different systems? So that remains to be determined. I'm cautiously confident that will turn out to be right, but science, of course, is ultimately proven by evidence, right? So that's research left to do over there. At the same time, we have taken advantage of the knowledge that we've gained in the system to design artificial systems that can cross the cell membrane with incredible efficiency, more efficiency even than the lateral systems. So, for example, we developed what is called a cyclic cell-penetrating peptides, which you mentioned early on. That's in our website. And those are the molecules that are designed to be very stable metabolically. That's good for being a drug. You don't want your drug to be destroyed by the body, in a very short time. And so those are the molecules we developed. They are very efficient in getting into the cell, and they can take drug molecules into the cell. And this is already commercialized. And there's a company that I co-founded about 10 years ago called Therapeutics. And that currently is taking 3 to 4 drugs into clinical trials. And so that's going very well. So we'll move from fundamental research in the lab all the way to commercial space. And hopefully, in a few years, patients will actually benefit directly from our research efforts.
Protein-protein interaction inhibitors. Tell us what this means.
Sure, yeah. So when I want to talk about undruggable targets, so protein-protein interaction, what we call the PPIs, those are the classical examples of undruggable targets. So here you have 2 proteins function by interacting with each other. And that's how they perform their biological functions. And when you have 2 proteins bind to each other, they're very difficult to interfere with using a small molecule. And so small molecules usually work as a drug. They work against targets, usually proteins, that already have a little cavity on them because small molecules are small. They don't bind to most proteins with enough affinity. So if they're going to bind to a protein with high affinity, the protein has to have a little bit of, cavity or pocket on them. And so only 10% of proteins have that. And so for most proteins, especially proteins involved in PPIs, they don't have such a pocket. So small molecules can't do the job. And so we need a large molecule like a protein, specifically, proteins like antibodies, and that can bind to essentially anything. And so if we can get large proteins into the cell, they would be bind to one of the 2 proteins involved in PPI, and then they can interfere with a normal PPI and therefore modulate the cellular activity and potentially act as a therapeutic agent. But when you have these large molecules, of course, come back to the question of how do you deliver them into the cell, right? Mm-hmm. So that comes back to penetration and the mechanism by which how things get into the cell. So this is why all of the things happen in my lab, they are related.
Another area that you're working in is the development of intercellular biologics and chemical probes. What's a chemical probe?
So intracellular biologics intended to be a new class of therapeutic agents, which is basically large proteins and other large biomolecules delivered into the cell, then they can act as a therapeutic agent. Chemical probes, meaning we can also use the same types of molecules as a research tool And that research tool can allow us to ask specific questions. Okay, so what is the function of this protein inside the cell? And if I use a chemical probe to eliminate the function of that protein, and then the cell should have a phenotype, right? We can look at the phenotype. Well, look at what cells what happens to the cell if we lock out the function of a particular protein, and that can tell us a lot about what is the biological function of the protein of interest. So the same kinds of molecules, the larger molecules delivered into the cell, can also act as incredibly powerful tools for us to study additional biological mechanisms and understand fundamental biology behind the living system.
So as you've been talking here, the question I keep asking myself is, are you a physicist or a chemist or a biologist, or maybe all of the above or maybe none of the above?
So that's a great observation you have made, David. Science today has become very interdisciplinary as well as multidisciplinary. So for drug discovery, it's truly a combination of many different fields, right? So there's chemistry, there's physics, there's biology, there's medicine, and there's even economics involved in that, because ultimately you will make a drug, you have to be able to make a profit, right? So it's really multidisciplinary. So my training was in organic chemistry. It's one branch of chemistry. And then, from there, I branched out into biochemistry, cell biology. And of course, today I also branch into medicinal chemistry, drug discovery. So I call myself as a chemist.
Why did you settle on chemistry? Because you were trained in organic chemistry? That's just what you sort of settle on, or?
Well, that's.
And you're in a chemistry department, I suppose.
I'm in chemistry department. That is true. If I look at everything we do, chemistry is the foundation of everything we do in the lab. So I think I call myself a chemist is probably the most appropriate.
Before we started recording, you and I were talking about research methods, the reasons for research, and you said something to the effect of there are two reasons or two drives, I suppose, for research: societal needs and curiosity. Say a little more about that distinction and maybe what drives your own research agenda.
Yeah, so I would see a lot of my colleagues, they do research, mostly driven by curiosity, right? So there are a lot of, let's say, biological phenomena or chemistry, physical phenomena that are not completely understood. And so, so people do research, try to discover truth, right, scientific truth. And that's science. It's what scientists are supposed to do, right? But then at the same time, there are true societal needs. And so what's driven me, the two things, right? One is the need for treatments for, many diseases. I mentioned earlier, there are lots of diseases that do not have proper treatments today. I'm particularly interested in genetic diseases where there's really no good solutions other than intracellular biologics that we mentioned early on. And then I'm also getting interested in recent years for sustainable agriculture. And now you may say, well, medicine and agriculture, how do they have in common? Well, actually, they have the same thing in common, is that in the past, even to a large extent at the present, the agriculture heavily depends on chemicals, pesticides and insecticides and things like that. And they are being effective. But, when we dump, millions of tons of chemicals into the environment each year, and these things can stay there for quite some time, and eventually, that's going to do a lot of harm to the environment. And so there's a lot of push recently from governments, particularly in Europe, for something that's more sustainable, less impactful on the environment. And so one of the directions is moving from small molecules to biologics, larger proteins, because the proteins can be degraded shortly after they're applied and then degrade into amino acids, which are natural components. So they have essentially no impact on the environment if we do them properly. Mm-hmm. And so we also become very interested in that. In fact, one of my OSU colleagues and I co-founded another company called Celta Agritech, and we're taking advantage of the technology we developed here, delivery of large molecules into the cell, and try to generate products that hopefully will benefit not just the sick people, but everybody in the society. and by providing, high quality foods that's free of contaminants. And so the idea is very simple, is that we're going to take a protein and we're going to deliver into plant cells. Maybe we're delivering into fungal cells. Fungi cause a lot of plant diseases, right? Mm-hmm. And then we can hopefully prevent fungi, from replicating in plant cells. And that would be a, wonderful type of agricultural product.
These two drives for research, societal needs versus curiosity, were you always driven by societal needs, or did you begin as someone who had a natural curiosity and you've moved toward an interest in societal needs?
That's a great question. So I try to combine the two. I'm trained as a scientist, so naturally I have a lot of curiosity. I'm interested in how things work. And scientists, in the academic world, I think making fundamental discoveries is still highly regarded. And because these are the, things that can have a long-term impact on human, society, right? So making fundamental discoveries is certainly very important. And I'm, very interested in doing that. So what I try to do is to do both. And look at the societal needs and ask the question, why is that people have not been able to solve this problem? And usually, it's because there's a fundamental piece that's missing, something that people do not realize. And certainly that's true in our case, right? These bigger molecules across cell membrane by this VBC mechanism, which is something that people never thought about. And so the discovery, the mechanism, the VBC mechanism is a fundamental discovery, and that hopefully, I hope one day we'll get into textbooks, right? So that's purely curiosity-driven type of research. But at the same time, once we understand this mechanism, it is incredibly powerful in terms of driving technologies. And so we're able to develop several powerful technologies based on this unique mechanism. And so that, and then allow us to solve societal problems.
What got you interested in chemistry? Or I guess maybe another way of asking this is, why are you a chemist as opposed to, a biologist or an historian or, a basketball player or something? What got you into chemistry?
Even when I was very young, I was always, curious about how things work. So getting, into science, it was pretty natural for me. And I was pretty good at it. I'm interested in stuff. I like to think about things, and I like to fix, out how things work. And so getting into science is natural. Getting into chemistry perhaps is more, perhaps by accident. I guess I did it very well in chemistry in high school. And when I was going to college, I really didn't know. I got to college in the early 80s, so that's the time before internet. I didn't know what's out there, right? So you kind of do things that are more familiar with. I was very good at chemistry. I was interested in it, so I thought, let's do chemistry. And then, of course, in college, and I fell in love with it.
You've mentioned that you founded at least two companies. Maybe there's more you founded. But I wonder, when you were starting off in college, when you were starting off as a chemist, did you imagine yourself as a founder of a company?
Oh, I never thought that I would be a founder or co-founder of a company. That's not until I went to graduate school, I would say. Yeah, so in graduate school, then I realized that, research can actually be directly used in the commercial space, because my former advisor, that's Professor Peter Schultz, who is currently the president of Scripps Research Institute, who was a phenomenal scientist. He's a phenomenal scientist, but also a very shrewd entrepreneur, and he started many companies and become very successful in that space. And so we saw how one can take a fundamental discovery from the lab into the commercial space.
Is this an expectation now for scientists, that in addition to being researchers, that they're to be entrepreneurs?
I wouldn't say that should be an expectation. I think if someone can make truly fundamental discoveries that can benefit the human society, that's fantastic. That's, if you can do that, that you are doing better than most people already, right? But I would say, to people who are interested in, taking their research into the commercial space, give it a shot. It's not guaranteed to succeed, but it might succeed. And it is very satisfying when you can take a research to the commercial space and make products out of them and see your research directly benefit people in some ways.
Is this how you teach your students, your graduate students, the postdocs in your lab?
That is certainly true. And actually, as a result of that, many of my students directly go from my lab, to pharma companies, to biotech companies, and, continue the kind of research to do here.
Tell us what's next for your research.
As I mentioned earlier, the VBC mechanism has been demonstrated in a few systems. I would like to demonstrate that is also applicable to other systems, so that will keep us busy for the next 10 years. I probably got 10 more years in my tank. And at the same time, I mentioned that I actually studied 4 companies. And so, I would like to see the companies succeed and then generate a few products that will benefit, the people with diseases. That's the therapeutic companies. And also, hopefully, we can develop some agricultural products that can benefit everybody. For example, around my home, we have fungal problems with my lawn. Oh. And so I hopefully, some of the products will be able to deal with the fungal diseases that's affecting my lawn.
Dehua Pei, thank you.
Thank you, David.
Voices of Excellence is produced and recorded at The Ohio State University College of Arts and Sciences Marketing and Communications Studio. More information about the podcast and our guests can be found at go.osu.edu/voices. Voices of Excellence is produced by Doug Dangler. I'm Jen Farmer.